Hello, Indiana Virtual. The purpose of this video is to give you a brief introduction to the lesson 0307 Biotechnology. So we will open up the lesson and like always, scroll down to the bottom of the first page and open the biology notebook. And I am going to go ahead and go to our Google site and open the biology notebook there. Okay, I actually already opened it. So what we are going to learn about in this lesson is genetic engineering, transgenic organisms, cloning, DNA fingerprinting, biological relationships, and wildlife conservation. Okay, so now you have an idea of what sorts of topics are going to be included in this lesson. Let's also give a little introduction here as to what else you will be working on here. Okay, in this lesson, you will learn how technology helps to compare and study genes and proteins. You will be introduced to the process of DNA fingerprinting and perform a virtual lab on gel electrophoresis. You will be able to describe the impact that DNA fingerprinting has on an individual and society and the impact biotechnology makes on individuals, society, and the environment. Okay, we'll come back and talk about what the actual assignment is here shortly. Okay, so now that we have the notes page open, now we have an idea as to what this lesson is about. We're going back to slide 1, and you're going to read the introduction here on biotechnology and learn about the glowing cat. This is kind of an old topic, so maybe you have heard about this otherwise. So anyway, read through slide 1, and when you feel you're done with slide 1, we are going to go on to slide 2, and you can make note here as well. There's also 5 slides in this lesson. Okay, our next topic is about genetic engineering. So we're going to learn what genetic engineering even is, what genetic engineers do, and how that sort of topic even works and how it applies in our lives. Okay, so you will learn about restriction enzymes, something else called DNA ligase, recombinant DNA, and that is what it kind of sounds like, DNA that's been combined together from different organisms. So you need to learn about how that actually kind of happens. Okay, and when recombinant DNA has been created and put back into a plant or an animal, or a plant or animal's DNA has been changed or recombined, then we've created a transgenic organism. Okay, and that's our first sentence here. Organisms with traits produced by recombinant DNA are transgenic. Okay, and recombinant DNA, genetic engineering, transgenic organisms are much more ingrained in our lives than what you think. Our crops all out there are genetically modified. So you'll learn about that. So we have a little interactive portion of the lesson here. Not super interactive, but you have details on slides that you're going to read about how our crops have been genetically modified through recombinant DNA, transgenic organisms, genetic engineering. And not just crops, but animals too. So you will continue to read about those, make your way through all of those slides, and then don't forget to read this little paragraph down here at the very bottom as well. Okay, when you are ready and you've documented everything you need to, go on to slide 3. And now we'll talk about cloning. And our first organism that was successfully cloned was Dolly. So we have a little sheep here. Dolly back in 1997 was produced through cloning, was created through cloning. So you'll read and learn a bit about that. And then you'll go on to DNA fingerprinting. Okay, and so DNA fingerprinting is it's a lab, it's an activity, a way in which we can analyze DNA to see what genes are found within a chromosome or within an organism. So it's actually a really cool experiment to do. I wish it's something that we could do in person. So there is a virtual gel electrophoresis lab on here, so you can click on that and go to that site and complete that DNA fingerprinting experiment through gel electrophoresis. Here are some images that show you kind of how that gel electrophoresis works. Okay, if you're interested in forensic science, gel electrophoresis and DNA fingerprinting is used all the time to identify DNA that was left at a crime scene and see if it matches potential suspects and such. And you have a did you know here. Okay, if you do this gel electrophoresis activity, then you might come across some equipment that you see in your CSIs and NCIS shows pretty often. Okay, when you are ready, go on to that next slide. And now you will learn a little bit about biological relationships and wildlife conservation. Okay, so now we have come to the last slide, and as you know, this is where your assignment actually is. So you can read through the final summary information, and then our assignment is... Okay, sorry about that. I got stumped a little bit where our assignment is. Okay, so this assignment, it is a short answer assignment, actually. So I am going to switch over. You can see there's no links or anything on here. This assignment actually is on the next page. It's what would look like a multiple choice assignment, but it's not a multiple choice assignment. So I'm going to show you a little bit here over on our Google site as to what this assignment is like. Okay, so I've opened up the assignment on our Google site, and it says it's a biotechnology writing assignment. Okay, now right here, this very first line is quite important. Choose one of the following scenarios and answer the questions provided. Support your answers using scientific reasoning and discussing possible impacts to individual, society, and the environment. Be sure to discuss any medical or ethical issues that may apply. And this writing assignment is worth 10 points. Okay, you have 2 scenarios to choose from. Again, you only have to pick one. Okay, scenario 1 is about genetic engineering and using genetic engineering to create more productive strains of farm animals. Okay, by adding genes to an animal's DNA, the animal can be made to produce milk or meat containing vital nutrients that would not be found naturally in those products. This provides a greater amount of nutrients per serving of milk or meat. Okay, so you have 2 questions to answer. Do you think this type of genetic engineering should be pursued? Explain your answer. Okay, what are some possible impacts, positive and negative, of this type of genetic engineering on individuals, society, and the environment? Okay, so maybe that topic, that question doesn't really interest you. Okay, you have another option here, scenario 2. You could answer this one instead. Both are worth 10 points. Okay, so scenario 2. In the future, doctors may be able to evaluate your DNA to check for genetic markers related to specific medical conditions or harmful reactions to medications. Your personalized genetic profile could be kept on record and referred to in order to develop personal medical treatment based on your unique DNA sequencing. Scientists have found that just small variations in a specific section of your DNA can help determine how well some medications can work for you or if they would be beneficial at all. Okay, so here you have 3 questions to answer. Okay, 1, would you want to have your DNA profile kept in your medical record as a reference for your doctors? Explain your answer. So yes or no, and explain. Question number 2, what are potential pros and cons of having such tests done and referring to DNA sequences when determining a patient's medical treatment? And 3, describe how the availability of this technology might affect the frequency of genetic diseases in individuals and populations. Okay, so this time with this assignment, you do not have to copy this as a Google Doc because what you'll actually do when you click on the Start button here, you'll see that there's scenario 1 and then there's scenario 2, and then you'll type your answer in the box provided. And then once you're done, you will submit your answers. Okay? So this assignment, it is just a little bit different as to how it's set up. Okay? But basically, it's a short answer assignment. Make sure you address all parts of whichever scenario you choose. You only have to pick one. Okay? If you have any questions, please reach out. Okay? Bye, guys. I look forward to hearing what you have to say. Okay? See ya.